A muscle consists of many different parts. The entire skeletal muscle is connected to the bone by a tendon. Within the muscle, there are small cylinders called fascicles. These fascicles are each made up of muscle fibers, also known as muscle cells. Within the muscle fibers are smaller cylinders called myofibrils. Each myofibril is made up of sarcomeres, which consist of thick and thin filaments. These sarcomeres are what pull together and allow the muscle to contract. Sarcomeres are separated by Z-discs. A thick filament is made up of myosin, while thin filaments are made up of actin, troponin, and tropomyosin. Now, when a person decides to move, the signal from their brain is sent down their spinal cord, which starts the contraction and relaxation cycle. This signal then arrives at the neuromuscular junction, which is the synapse formed between the axon terminals of the motor neuron and the motor end plate of the muscle fiber. This nerve then triggers a muscle action potential. The synaptic end bulb of the neuromuscular junction proceeds to release a neurotransmitter to the motor end plate. In this case, the neurotransmitter is acetylcholine. Next, the neurotransmitter binds to the muscle fiber. This triggers the sarcoplasmic reticulum to release calcium. Then the calcium binds to troponin, which moves the tropomyosin off the actin binding sites. This allows the myosin to bind to actin. When the myosin binds to the actin, it releases a phosphate group, ATP. Next, the cross-bridge swivels, releasing ADP. This shortens the sarcomere. The ATP then binds to the myosin, which causes it to release from the actin. The ATP is then broken down into ADP. This activates the myosin head to bind and start again. The latter process is called the sliding filament model. To be more specific, the sliding filament model is where myosin heads bind to actin binding sites. When this happens, the myosin pulls and slides the actin and Z-discs towards the H-zone. As a result, the I-bands and H-zones become more narrow. This sliding generates force and shortens sarcomeres and thus the muscle fibers. The contraction cycle repeats as long as the calcium concentration is high. Then, to relax, the acetylcholine must break down to stop the muscle action potential. The calcium ions are then transported back into the sarcoplasmic reticulum. Because there is no longer calcium to attach to troponin, the tropomyosin covers the actin binding sites, keeping the myosin from binding to the actin. With this process, the muscle relaxes. And that is the contraction and relaxation cycle of a muscle.